scusate, perché ho fatto comunicazione che effettivamente non è necessario che sia io che parlo, perché il link del sito no,
YouTube, è YouTube adesso.
YouTube, io avevo provato a chiedere anche ad altri.
No, ma l'abbiamo imparato anche noi 3 secondi fa, quindi
scusate, sì, allora niente,
scusate, ciao, grazie a te.
Prima di iniziare volevo fare una introduzione di non più di 40 minuti. Vorrei tanto che questo evento fosse un pochino più ufficiale con il pubblico, eccetera. L'abbiamo adottato per venire incontro a delle raccomandazioni ministeriali, ma vi assicuro che, almeno da parte mia, ma sono sicuro anche da parte dei colleghi, c'è un genuino interesse, siamo contenti di essere qua per ascoltare, perché non sempre possiamo ascoltare le presentazioni di Televaiva di tutti quanti, e quindi volevo ringraziarvi per essere qua e per raccontarci le storie che avete sviluppato negli ultimi 3 anni. Tutto qua, grazie.
Okay. So good morning. First of all, thank you for the opportunity of presenting my PhD project named Boosting Anti-Cancer Immunity with the help of genetically engineered stem and progenitor cell transplantation. As you know, cancers are comprised not only of cancer cells, but are infiltrated by different cell types, namely the vasculature, cells of mesenchymal origin, and a large array of hematopoietic origin cells, so cells of the immune system. Among them, macrophages have been discovered to infiltrate tumors, and they integrate different signals and between cancer cells, but also cells of the immune system. And usually, their presence within tumor tissues correlates with dismal prognosis in patients. Importantly, these TAMs, tumor-associated macrophages, can be targeted for therapeutic purposes, namely, especially by depletion strategies or re-educational strategies. And there stems in a platform that was developed in our institute by Luigi Naldini's lab. Early works demonstrated a subgroup of these TAMs, namely type-2-expressing monocytes, that I will refer to as TAMs from now on, which circulate at low frequency in the peripheral blood of humans and also in mice in preclinical models, and have been shown to infiltrate tumor tissues. They are a pretumoral TAM subset with perivascular localization, and their role in tumors is mainly mediating angiogenesis and immunosuppressive function. In preclinical models, their genetic ablation has been demonstrated to curb tumor growth. Thereby, in the institute, they have developed a strategy to exploit their biological functions to deliver transgene of interest, in particular pro-inflammatory cytokines, by means of a regulated cassette introduced by the antiviral transduction, regulated at the transcriptional level by type 2 promoter enhancer sequencing, and at the post-transcriptional level to avoid expression in the more primitive hematopoietic stem and progenitor cells, which would have type 2 promoter enhancer activity. Thereby, this therapy is deployed by means of autologous transplantation after ex vivo transduction of hematopoietic stem and progenitor cells. At steady-state conditions, all the hematopoietic progeny does not express the transgene of interest, but when a cancer is present, these cells are physiologically recruited at tumor tissues, and there they switch the transgene cassette on, achieving a local release of the cytokine of interest in the tumor microenvironment. Preclinical testing of the TAM-based delivery of interferon alpha have been developed in the institute over the years. These are just some examples of anti-cancer efficacy in different tumor models, such as a glioblastoma tumor model, breast cancer metastasizing to the lungs, or colorectal cancer cell line metastasizing to the liver. These preclinical studies and also biodistribution and safety studies paved the way for the clinical translation. Indeed, nowadays there are 2 phase 1-2 dose escalation clinical studies open at San Raffaele in multiple myeloma and glioblastoma multiforme, sponsored by Genetic Science. And here stands my PhD project, and in the first part I will talk about this strategy, delivering cytokines other than interferon alpha in a mouse model of Vita-Salute lymphoblastic leukemia. So first of all, we cloned into the same cassette the cytokine of interest, the interferon gamma or TNF alpha, and with transduction at clinically relevant doses between 0.4 and 1.2, we achieved long-term transplantation of the cells, and engraftment of the cells is exemplified by the CD45.1 expression donor cells, without evidencing hematological, overt hematological toxicity in all the lineages. Thereby, our strategy is safe by delivering these cytokines by this platform. Then we tested the efficacy of this therapy against a murine DLL model, which has been developed in our lab by forced microRNA 126 overexpression in murine negative cells. And as you can see, against different murine diseases, we had a robust anti-leukemia effect, especially early time points with interferon gamma. This was not the case so evident for TNF-alpha. And indeed, we went on to characterizing again the efficacy of interferon gamma against another primary disease. And then we wanted to detect whether the immune system was mediating the effect of the efficacy that we detected. And to do so, we did transplantation in the same setting, but in this case, we also transplanted donor cells devoid of the interferon gamma receptor 1 receptor. So the immune cells were not able to see the interferon gamma that cells were secreting. And at a vector copy number between 0.2 and 0.6, we detected anti-tumor efficacy when the immune cells had the interferon gamma receptor, but not when they were devoid of it. And the signs of efficacy were also accompanied by signs of immune activation, such as MHC class 2 upregulation and infiltration of cytotoxic T lymphocytes. Thereby, the immune system is an actor that mediates the efficacy of our gene therapy. To study this more in detail, we conducted single-cell RNA sequencing at 2 different time points. In early time points, when the anti-leukemia effect was more evident, and at later time points when we displayed some loss of efficacy. And we were able to detect all the immune cell populations in the bone marrow of mice, in particular different clusters of T cells and different clusters of myeloid cells. Here I highlighted the M2-like macrophage populations because when we interrogated our myeloid dataset with a 10-base signature derived from the literature, we saw that the higher expressing cells that resemble at the transcriptional level the TEMs are indeed the M2-like macrophages. We also coupled to gene expression profiling by single-cell RNA sequencing PCR, single-cell PCR sequencing, and interestingly, we found that just interferon gamma-treated groups, especially day 12, exhibited expansion of clonotypes, meaning that clonotypes represent between 1 and 10% of the sequence TCR. And in particular, these clonotypes fell within the cytotoxic CD8 cloud by transcriptional activity. Indeed, these data suggest that not only interferon gamma gene therapy increases the infiltration of TCRs, but also favors the clonal TCR expansion. To gain more insight into the loss of efficacy, we compared the gene expression enrichment analysis at day 17 versus day 12. And first of all, we saw that in the disease, which is this column here, we saw an upregulation of marker genes associated to more primitive disease, more aggressive disease. And in general, as you can see from these rows, which are hallmarks of interferon response, we saw a loss of responsiveness to the cytokine that was delivered, indeed, still delivered, and we detected it also by the M2-like macrophages, which were the ones with more resembling the TEMs by genetical activity. And therefore, we wanted to combine our gene therapy with other cytokines or immunotherapies to prolong the anti-cancer activity. That's what we did, first by combining different cytokines, namely interferon alpha, interferon gamma, interferon alpha or TNF-alpha, and especially interferon gamma and TNF-alpha. And so that the more robust anti-leukemia activity was obtained once we combined interferon gamma and TNF-alpha, and this was associated again with signs of immune activation, the same that I depicted before. And interestingly, when we combine instead our gene therapy, which are the green groups, with immune checkpoint blockade O1-MT, which is an inhibitor of IDO, we saw that, first of all, checkpoint blockades, as expected, gave rise to broad, very variable anti-leukemia responses that were, to some level, increased when we combined it with our gene therapy. 1-MT combination led to more homogeneous kind of response, although none of these mice really had very, very low levels of the disease. And the activity, again, was correlated with signs of immune activation, such as MHC class 2 upregulation of myeloid cells and infiltration of cytotoxic T lymphocytes. These were obtained in B-cell acute lymphoblastic leukemia model, But then in the second part of my PhD, I also investigated the role of interferon alpha gene therapy in a multiple myeloma, because multiple myeloma is a disease that affects the immune system, and we wanted to study it also in biobank materials of patients treated at OSR longitudinally. So patients in a retrospective cohort of patients were sampled at diagnosis after induction therapy with a triplet-containing regimen, post-transplantation, or eventually at relapse. We decided to choose patients based on two categories of them. So one group of patients displaying a relapse after transplantation, and one achieving long-term remission. As you know, the immune system is really a hot topic in myeloma nowadays, as evidenced by recent publication by single-cell sequencing analysis of immune cells enriched in the tumor microenvironment compared to normal bone marrow. We thought authors showed recently that myeloid cells, especially the CD16 positive myeloid cells, monocytes, are enriched in the tumor microenvironment and some subsets of T cells. And concerning myeloid cells, what we saw was an upregulation, a slight upregulation of myeloid cells after induction therapy. And in particular, the CD16 positive monocytes, the intermediate non-classical monocytes, were upregulated in the early relapse patients compared to the long-term remissions in a time point after transplantation. Concerning the lymphoid compartment, we saw this was known in the literature that transplantation led to a dramatic impact on the T cell compartment by skewing the balance in favor of the CD8 compared to the CD4, and also reducing the more primitive, naive, and central memory population of T cells. And indeed, this was also evident that when we split our patient cohort, because some patients, early relapse patients, displayed an increase the effect over T naive ratio in the CD4 compartment after transplantation. We also studied flow-related immunosenescent markers, which are loss of CD28 and acquisition of CD57, and this phenotype in CD4 compartment especially increased over time and over different line of treatments, and in particular within the CD4 cells, the senescent CD4 lymphocytes were increased in early relapse patients compared to long-term remission patients as early as post-induction, after induction therapy. So we wanted to study the effect of interferon gene alpha gene therapy in a immunocompetent mouse model, and we first investigated it in the VTMX mouse model, which is a spontaneous and stochastic model of multiple myeloma. Importantly, this model has predictive value for clinical efficacy of anti-myeloma drugs, and the progression of myeloma correlates with time accumulation. So in this case, we designed therapeutic intervention experiments, meaning that when we detected an M spike, we irradiated just the lower limb, not to abate completely the disease, and then we transplanted our gene therapy and followed the disease over time. As you can see, when we split the treated mice in those with a as high as 4% of chimerism, which is rather low, we had a stabilization of the disease in most of the mice, and some mice, here you see that are the responders, which were increased compared to the mock treated, and a reduction of the dead mice over time. This was correlated with the reduction of plasma cells in the bone marrow of both the irradiated limb and also of the non-irradiated limb, so the upper limb, compared to the mock treated mice. And this activity was accompanied by expansion of T cells, infiltration of T cells in the bone marrow, both the CD4 and the CD8. Then we also wanted to characterize in a humanized platform the really interferon alpha construct against myeloma, human myeloma cell line, and we did so by first transplanting the CD34 positive ex vivo transduced cells, and then injecting the myeloma cell line. And following the disease over time, we saw over different experiments that by whole body bioluminescence and by flow cytometry that the treatment correlated with approximately 50% disease reduction compared to controls, and this was accompanied by signs of immune activation against, more importantly, skewed in favor of the CD16 positive monocytes and upregulation of HLA in bone marrow monocytes, and increasing the proportion of bona fide dendritic cells in the bone marrow. So we wanted to combine, given the signs of immune activations, our gene therapy with other immunotherapies, but not only, also because multiple myeloma is a disease that is treated with combinational strategies. And as you can see here, on the left is the combination with the anti-CD38 monoclonal antibody, daratumumab, or proteasome inhibitor, or immunomodulatory drug lenalidomide. In general, we did not find overt synergism, but in all the cases, we found that the more robust anti-myeloma effect was obtained with the combination of the two treatments, and we did not observe for neither of these combinations overt toxicity in all the treated mice. And lastly, to push the system even further, we combined the same platform with a cell therapy, with NY-ESO-1 with directed T-cell transfer. This was done in collaboration with Professor Bonini's group, with Vita-Salute, and first T-cells were isolated, stimulated, they perform a tract disruption, and they introduced the novel TCR by nuclear lentiviral transaction. And importantly, they also differentiate, they kept the cells in culture to preserve the T cells and memory and central memory subsets. And against what was the activity against myeloma disease, the MM1S is a very aggressive disease. So basically, the disease grew over time, and with vector copy number as low as 0.25 in culture, we saw that the TCR groups displayed a more robust anti-myeloma effect, which were more evident by combining the two treatments. And against another myeloma cell line, more indolent, we saw that the disease was abated over time, but the kinetics were faster when we combined the two treatments together. So to conclude, I presented to you the safety and effectiveness of our gene therapy platform, which is amenable to deliver different pro-inflammatory cytokines other than interferon alpha. We demonstrated that there is a modulation of the tumor microenvironment, especially in the immune system, which is different between interferon alpha and interferon gamma, and it allows safe and effective combination with other therapies. This platform is amenable for many oncological indications, importantly, regardless of target antigen expression. And indeed, these studies, not only, of course, have paved the way for the translation, which, I repeat, are ongoing today at Ospedale San Raffaele in the context of multiple myeloma and glioblastoma multiforme. So importantly, I would like also to thank, first of all, Bernard Gentner for allowing me to work in this project for all these years, and Adele and Francesco and all the lab members, the hematology unit in particular, Professor Fabio Cittari, Professor Naldini, and all the collaborators, and of course you for your attention. Thank you.
Okay, well, thank you very much. Congratulations for the massive amount of work that you did. And I wanted to ask you, in respect to this, how much of this work you basically mastered? Although I know it's a very collaborative effort, but I wanted to ask you what you did mostly by yourself and what you did in collaboration or with the help of other colleagues.
Yeah, okay. So, of course, collaborations are today essentials, and we did a lot of them. What I mastered more are especially the in vivo experiments and all the handling from the idea of a novel, let's say, experiment and to the endpoint analysis, let's say. Of course, the single cell analysis, which is a large chunk also of the project, was conducted with bioinformaticians especially. So we instructed biological questions, but then the codification and all the stuff was implemented by them and also by our lab, some of our lab members. And basically, these are the two areas that I did the most, let's say. For the myeloma cohort, the flow cytometry data, the identification of patients, that was something that I was, let's say, more acquainted to because of my previous work studies. And the combination with the T, of course, with Professor Bonini's group, that was a very fun collaboration. We performed also other collaborations that I didn't have time to mention. Yes, of course.
So, where would you be moving from here? What would be the next steps of your already remarkable achievement?
Today I started, as of now, so I can answer. I started the specialization because I decided, I think that doing a medical specialization is essential in today's also scientific world. So I entered into the specialization in medical oncology, and I hope, so now I open more the focus on that side, but I hope that once I get, let's say...
Finish your residency, then you will be able to do both.
Yeah, also because, but once I know how to manage also that, I realize how to move the next step.
Which university?
Statale, I'm at Yale now. European Oncological Institute, Yale.
Okay. One question regarding the effect on the acute leukemia.
Yes.
Most of the results seem to be quite bimodal, meaning that there is a population that is really recovering well, while others are really down to background. Did you see or did you separate those, for example, with the Zinc effect to see what is the difference between the responding and non-responding?
Yeah, thank you for the question. Yeah, that's something very that we saw, especially with the combination with the checkpoint blockers. Yeah. And in that case, this is something that is, let's say, that we in a way expected in some animals, because immunotherapies are highly variable. Sometimes there are some very good responders and some that do not respond very well. By single cell, we saw this loss of responsiveness to interferon gamma. However, we're not able to find gene expressions associated with, for example, precise desensitization or the signaling from the interferon gamma receptor that went down. So we did not detect a precise mechanism as of now, but this is something that we are still asking ourselves from those analyses. Yes. Also on the T-cell side, for example, yes, not yet found something.
There's a question from Professor De Curtis.
Can you go to slide 6? I ask you a comment because I am not responding. About the time frame we were using in this first part. Yeah. You can go out and choose the slide. I think we can also just discuss. Can you exit the presentation? Not respond?
The mouse, no.
Okay.
Okay.
Okay, now it's going down. It was about, so yeah, so because I in the numbers, yeah, there was you were showing that there was an effect at early time points.
Oh, yes. Yes.
Okay, and then there is but then there is sort of a rescue in a way. This is one example of this on the left graph. So this time frame is very short, and then there is a very quick rescue. So how is this? Can you comment on this? How could this be then effective on the action of this?
Yes, first of all, this is due to the disease kinetics. We think that up to some point, the immune system, which is activated eventually by interferon gamma, is able to cope with the leukemia cells, also numerically speaking in a way. But at some point, there is a threshold by which the leukemia is so large enough that it progresses more quicker, let's say, than the immune system is able to. And also, by single cell, we were able to identify some, especially day 17 compared to day 12, some of primitive markers and also markers of proliferation. So it seems that the leukemia itself is selected towards a more aggressive form of the disease. So both of the two aspects, I think, are going on.
I have some more translational questions. Since you have a medical background and you're going to start your residency in medical oncology, how would you envisage to translate these strategies in the clinical setting? So both for ALL and multiple myeloma, what do you see? So this would be a transplantation of hematopoietic stem cells from an allogeneic donor, from an autologous donor. How would you couple this with of immunosuppressive agents? So can you do that?
Yeah, that is a very interesting question. It depends on the disease, first of all. I think that myeloma was also ideal because the autologous transplantation there is a state of the art. And especially I would envision immunotherapies in general, and also this one in early stages of disease in general. This is not easy to have indications for that, also for clinical trials, but I think that going in that direction is a must do, let's say. Also, I envision it by combining this therapy with other regimens. I didn't show also some rationale for combining with the CD38 is that the target antigen is upregulated by interferon alpha, so should work more. This is, and then this is, let's say, a platform. First of all, also it may serve to prevent metastatic spreading in different organs. Preclinical data have studied this, so it may also be useful in that setting. And then we can also think of changing a bit this platform, going to more differentiated kind of cells to have, and it is something that also in the Institute of investigating. So I think it's very broad, and I would go in early phases and in general, but then each disease is different. So it's
okay, if there are no other questions, we can move on to the next candidate. And thank you very much for your brilliant presentation. Okay, the next presentation is by Filippo Birocchi. The curriculum is basic and applied immunology and oncology again. The title is Development of Gene Therapy Strategies for Inducible Regulation of Protein Expression for Therapeutic Purposes. The floor is yours.
Start. So good morning to everyone, and thank you for attending my PhD final public defense. So interferon alpha represents a valuable and promising agent for cancer immunotherapy because it attacks at different levels. So it acts on the tumor cell by inducing the upregulation of MHC class 1, for example, is able to reprogram the tumor-associated macrophages toward a pro-inflammatory state, can also suppress the activity of immunosuppressive cells like myeloid-derived suppressor cells and Tregs. It can also activate different effector cells like T cells, NK cells, and dendritic cells. However, its clinical application has been limited by the toxicity associated to its systemic exposure, in particular, for example, neutropenia, hepatotoxicity, but also depression symptoms later on. So, as you can see, for example, recombinant administration of a cytokine like interferon alpha give rise to these classic peak and trough kinetics followed by each administration. And if you consider this gray box as therapeutic window, very rarely the concentration resides in this time frame, and most of the time is above it, so in a toxic dose, let's say, or below it, so in a ineffective range. So gene therapy can solve this problem because it can give rise to a stable expression of the cytokine, which can reside in the therapeutic window for all the treatment. And also we can think of engineering this approach to be local, and so thus preventing the systemic exposure. But how can we do that? So in the lab in the past, we discovered a new class of monocyte macrophages, which are characterized by the expression of the type 2 receptor. These so-called TEMs are specifically recruited to the tumors, but also to metastases, are perivascular, and produce bronchogenic factors, so they elicit a pro-tumoral role. So the idea of the lab was to convert these tumor-associated macrophages into Trojan horses in order to deliver interferon alpha specifically in the tumor microenvironment, thus avoiding the systemic exposure of the cytokine. So how can we do that? We, in the past, developed a lentiviral vector in which the expression of interferon alpha is under the control of type 2 enhancer and promoter sequences. So by transducing a macrophagic stem cell and transplanting them, the type 2 positive progeny, so the TEMs, will produce interferon alpha and only in the tumor microenvironment. We decided to apply this technology to glioblastoma multiforme because it represents an unmet clinical need. And despite the available standard treatments, which are surgical resection, radiotherapy, and chemotherapy, the prognosis of the patient remains extremely poor. This is because of the strong immunosuppressive nature of the tumor microenvironment of GBM, but also by its pathological feature, like, for example, invasion, which limits the success of surgical resection. So to test our technology in a syngeneic mouse model of GBM, we chose the MGB2 model, which is a novel mouse model of GBM originated by the specific deletion of P53 and P10 genes in the neural stem cells. By sequential in vivo passage, they originated these cell line, which is implanted in the striatum of the animals, give rise to 100% of gliomas, which resembles most of the feature of the human GBM, including hemorrhages, necrosis, and also, as you can see here by this image, a very invasive pattern, so very similar to the human pathology. We first confirmed the presence of, in this model of our population, so the TAMs, as you can see by this double staining, the TAMs are present in this tumor. And indeed, we decided to test our technology in this model. How can we do that? We transduce a murine hematopoietic stem cell with a lentiviral vector containing empty vector as control, or T2 interferon as treatment group, and we transplanted them in laterally irradiated black 6 mice. After hematopoietic recovery, we injected the tumor, and we then followed the mice through MRI scans. So even in this very stringent setting, so 100% of transduced HSPC, which is a very high dose, we observe very limited effect in terms of toxicity. We see very mild reduction in white blood cell count, in particular in the B cell compartment. And we also observe interferon alpha in the plasma of the animals, but a level which is quite low, around the 50 picogram per ml. So what about the efficacy? So as you can see here by these 4 representative mice per group, you see that in the tumor, in the control, the tumor grow very massively, while in the interferon-treated animals, the tumor growth was reduced and in some cases completely abolished, as you can also see by this tumor volume quantification. The interferon treatment was also able to prolong the survival of the animals, and a small fraction of the animals were also able to completely clear the tumor and survive long term. As you can see here, the tumor starts to shrink and disappear completely. So based on this preclinical data and previous data from the lab, an ongoing clinical trial is ongoing in our institute for GBM patients with the drug called Tempiron, so based on this strategy. So I showed you before that gene therapy is good, but inducible gene therapy is even better. Because if we can control the expression of a transgene, we can tune the expression in the therapeutic window. And at the end of the therapy, possibly after tumor eradication, we can turn off the expression of interferon, then coming back to a physiological situation. So this very challenging possibility can be achieved in different ways. And we try to do it using the destabilizing domain-based technology, then applying it to our interferon gene therapy. So by fusing a destabilizing domain to your protein of interest, all this is transcribed and transduced into protein. But a steady-state condition, the destabilizing domain is recognized as incorrectly folded, and all this is sent to proteasomal degradation. So the steady state is off condition. But if you had a specific ligand which binds to the stabilizing domain, this is stabilized, and together with it, your protein of interest. So it's a very simple system, single component, so it's very feasible for a gene therapy approach. We decided to use a destabilizing domain coming from E. coli, the hydrofolate reductase gene, and its ligand is trimethoprim, so an antibiotic drug approved, which is known to cross the blood-brain barrier, so it's convenient for GBM. So we then cloned the DHFR both at the C term or at the N term of interferon alpha, and we found out that when we fuse a DHFR at the C term of the protein, we have a good inducibility in vitro, and also we perform a functional assay. We observe that the protein is functional in inducing interferon-responsive genes, although to a lower extent compared to the wild-type protein. We decided, indicating that possibly the DHFR is interfering with the functionality of the protein. We decided anyway to go on in vivo with this construct, so we performed the same experiment that I showed you before, but this time with interferon DHFR. We injected the tumor, and then this time, after 2 weeks, we started the trimethoprim treatment. So we are more in a non-prophylactic setting compared to the previous strategy that I showed you. So after induction, we observe a small reduction in white blood cell, but a lower extent compared to the standard strategy. And we also observe an induction in the plasma of interferon alpha of these mice. What about the efficacy? Even the inducible strategy were able to reduce the tumor growth compared to control. And you can also see here by tumor burden quantification. Interferon alpha DHFR was also able to prolong the survival of treated animals with a long-term survival, even in this case. And then it's remarkable to see that if we don't turn on the expression, so if we kept one group, if we keep one group in the off condition, the survival is identical to controls, indicating the tightness of the system. Instead, if we are in the on condition, we observe this prolonged survival. Even in this case, we observe a long-term survival, and it's going to be interesting to see the dynamic of tumor growth of this mouse. You can see that before the start of the treatment, the tumor was visible, then start to grow, but then the therapy started to be effective, and the tumor shrink up to complete disappearance. We then stopped the trimethoprim treatment, and even after trimethoprim withdrawal, the tumor did not recur. So we tried to perform tumor rechallenge on the other brain hemisphere, and the tumor remained tumor-free even in this case, indicating that we possibly mounted a complete immune response against cancer cells. We decided to stress even more this system by comparing with systemic administration of interferon alpha in a dedicated experiment. And these are the tumor volume in the different MRI. You can see that in the first MRI, by comparing recombinant interferon and gene therapy interferon alpha, you see that both strategies were able to reduce tumor growth, even if in not a statistical manner. But in the second MRI, this effect was completely lost in the recombinant treatment, while it was maintained in the gene therapy approach in a statistically significant manner, indicating the superiority of the gene therapy over the recombinant treatment. If we look at the effect in periphery, we clearly see that after induction, we see interferon alpha in the plasma of gene therapy treated animals, but the levels is much lower compared to the recombinant administration. So we perform another experiment in which we compare the gene expression in the brain, healthy brain and tumor, but also other organs in recombinant treated animals and gene therapy treated animals. If we look in the brain, in the healthy brain, we clearly see that by looking at different interferon responsive genes, but also cytokines involved in immune response, we have a strong induction of all these genes in the recombinant treatment, while no induction was seen in the gene therapy. If you look in the tumor, all the groups can experience an induction of every gene in the tumor microenvironment, indicating a more inflamed status of this tissue compared to the brain parenchyma, but the induction was higher in the gene therapy group. And if we compare the gene therapy towards the recombinant treatment, we clearly see the higher specificity of the gene therapy, local gene therapy over systemic administration. This is even more black and white when we look at other organs. You see that recombinant interferon is inducing interferon response in all organs analyzed in a specific manner, while the gene therapy has no induction or very little compared to recombinant administration. This indicates the higher specificity of gene therapy compared to systemic administration. We then dig deeper in the mechanism of action of our gene therapy approach, that we therefore perform single-cell RNA sequencing on tumor-infiltrating hematopoietic cells coming from 3 control animals, 3 interferon gene therapy, and 3 inducible gene therapy animals. So this is a representative human plot of the whole data set merged together. You can clearly see that the tumor microenvironment of GBM is composed by different cell population, including macrophages, T cells, dendritic cells, NK cells, and B cells. We then perform we clearly see that by looking at the differential expressed genes between interferon inducible strategy, interferon strategy compared to control for each cluster, we have the upregulation of genes that you can see in red belongs to interferon alpha response, indicating that we are effectively delivering interferon alpha locally. And we confirm this also by gene set enrichment analysis, that we have enrichment in genes belongs to the interferon alpha response. And most of all, the cluster of monocytes are the ones that respond more to the presence of interferon in the tumor microenvironment. While in the downregulated genes, we clearly see genes belonging to the macrophages compartment, and in particular, the most downregulated gene is CCL8, that I will show you later belongs to a specific cluster of macrophages. We then subcluster different cell population, but for lack of time, I will show you just a few of them. We subcluster T cells. You can see that there are different cell population of T cells, CD8, CD4, Tregs, naives, and proliferating cells. Interestingly, interferon and interferon-DHFR animals are negatively enriched in genes belonging to the exhaustion signature, so indicating that interferon gene therapy reduces the cell exhaustion. And we confirmed this also by tax analysis. So you can see that by looking at the co-expression of PD-1 and LAG3, the two non-inhibitory receptors in CD8, we have a reduction of this fraction in interferon gene therapy treated animals. If we look at the monocyte macrophages compartment, these are the three groups belonging to the control, interferon alpha, DHFR. What catches the eye is this cluster 6, present in control but completely absent in the treatment groups. This is also confirmed by the cluster cell composition. You clearly see that cluster 6 is composed by cells almost exclusively coming from control animals. What is this cluster 6? So cluster 6 expresses CCL8, so the most downregulated gene that I showed you before. Also, for example, MRC1, a known M2-like marker. And indeed, if we perform a gene set enrichment analysis with published signature of alternatively activated IL-4 signature or classically activated with LPS and interferon, we clearly see that cluster 6 is positively enriched in genes belonging to the M2-like signature and negatively enriched in the one of M2 signature, indicating that indeed are possibly M2-like macrophages. Instead, if you look at the cluster which is composed more from the interferon group, for example, cluster 2 here, you see that express markers of M1-like macrophages like interleukin-1 beta and SOX3, and indeed is positively, the marker genes are positively enriching genes belonging to this pathway. Lastly, we then focus on the role of this cluster 6 in the human GBM. So these are the marker genes of cluster 6. What we did is we derived a signature based on the top 10 markers of this cluster. And as you can see, this signature is able to distinguish two populations in the human GBM. So the ones that express higher level of the genes experience a reduced progression-free survival compared to the others. And by more specific analysis, we understood that MRC1, PF4, and CD81 are the ones that are the most important in this signature. So indicating that this cluster may also, this population may also have a role in the human GBM, a detrimental one. So to conclude, the time-mediated release of interferon alpha is able to reduce GBM growth in this model up to complete eradication and resistance to tumor challenge, is able to prolong the mice survival. This effect can be due to a complete reshaping of the immune microenvironment over a pro-inflammatory state. We also confirmed the superiority of the gene therapy over the systemic administration. And indeed, we found this population of tumor-associated macrophages, which has a negative prognostic value that completely abolished in the interferon treatment. Our inducible strategy, in addition, confirmed the possibility to spatiotemporally control a cytokine in the tumor microenvironment and confirmed the efficacy in a non-prophylactic setting. So I would like to thank my lab, in particular Luigi Naldini, my mentor, Nadia Coltella, who supervised me during my PhD, all the team that worked on this project, all the internal and external collaborators, in particular Stefano Beretta, the bioinformatician that helped me with the analysis with single cell, and all of you for your attention.
Very nice work. One comment is you always show white blood cells. I would suggest the presentation to show the population because my perspective would be what is really impacted by.
Yes. So yes, sorry, I usually put some time, but for lack of time I didn't. But usually when we observe a white blood cell reduction that affecting many populations, or most of all the lymphoid compartment, in particular B cells. So if we look at the absolute number, usually it depends on the strategy. If we're using the inducible one or the not inducible, we observe a reduction.
Sorry if I interrupt because you mentioned also Weinsteinian. I'm surprised because these cells are really minority of the white blood cell count. So to see such a big difference.
Well,
it's strange, but I mean, it's just to be sure that it's not let's say, a diffuse reduction in all the population. Maybe the disease is moving more. So
Well, for this point, there's quite a, let's say, difference between humans and mice. So in mice, at least in a irradiated transplant, we observe a very high level of white blood cells, higher than wild type, and usually there's a very big component of B cells. So we observe this reduction, which usually in the patients treated with the recombinant interferon is more affecting the myeloid compartment. So we don't know exactly if we can compare these on a translational point of view. We know that in our case, in irradiated mice and transplanted, which is not the physiological situation, we observe this reduction, which is quite significant. So usually it starts from 50–55% of B cells and going down up to 40%. So it's quite addressable.
The other thing is, again, earlier, that we see now with the bimodal response. So you mentioned some mice survive and other type of effect. So it would be nice again, the genetic modification to focus to really show also when you show the different single cell analysis again, but really to probably show those who responded and those who did not. You do see in age or striking difference. Otherwise, I think the effect will be.
Yeah. So regarding the bimodal, the tumor growth is really peculiar. So it's quite a slow tumor, which leads to death of the mice around 60 days after the injection. So there's a very long time in which the tumor don't grow much, and then start to explode. So you have this model in general, also in the control. Some mice start to develop this tumor very big. Regarding the effect, clearly, that's the point I think everybody wants to investigate. The big problem of this model is that we have to monitor the mice through MRI scan, and we have very few MRI slots per month, actually 2 slots per month. So picking the ones that will respond is very difficult, very challenging. So we are trying to move for luciferase model, in which we can maybe find the ones that are going to shrink and clear the tumor. It's not easy. I was thinking for an experiment for the future to perform maybe a longitudinal analysis, so to collect, for example, samples from the blood, and then later, when we know who developed the eradication, we can check if there's something changed. But for single cell analysis, it's very challenging, very challenging.
I have a very basic question. The cells that you transplanted, P53 and P10 null, how representative are they of the tumor and tumor environment in actual glioblastomas?
So yeah,
I know that they're standard, but we're likely to.
No, yeah, P53 and P10 genes are known mutation for the GBM pathology. Clearly, we didn't focus on the genetic features of this model, but the paper of our collaborators did, and apparently they harbor other mutations, spontaneous mutations, which are found also in the human pathology. So from that point, clearly we observe these similarities, but clearly it's a model, so it's it's much more, I have to say that it's much more resemble the human pathology compared to other preclinical immunocompetent model like GL261, which have a very bulky growth. So this is one of the big problems of preclinical model of GBM. This one is very infiltrative. And we also observe spontaneous necrosis, which is also seen in patients. So it's somehow
close representation.
Clearly, yeah.
Very good. Thank you very much. Any other questions?
I have a curiosity more than questions. So, did you measure vector copy number in the cell culture and in mice after engraftment with your product? And what
yes. Usually the interferon, usually the target of transduction is around 1 and 2 vector copy per genome. But usually when they go in vivo, they always reach 1. So they never go higher than 1. Possibly there's a negative selection for the cells that are highly transduced. So that's for sure. Indeed, when we go very high with the dose, we usually see a delayed engraftment. So that is one point. Yeah. Again, this is only in the full dose transduction. So this is something that is not happening in the patient, for example, in the ongoing clinical trial.
And I have another one, I don't know if it was feasible. So you showed your cluster sticks in some human samples from glioblastoma, which fell well with your results in terms of the gene expression. Did you have the chance, because patients have been treated with this strategy, I don't know whether there has been a chance after treatment to have a sample from a patient who did a biopsy or a surgery, and to check after treatment whether the expression was changed?
The Bernard Genter lab and Genento Science are working on that, so we hope soon to have a comparison. Yeah, we're really excited to do that.
Very good. And thank you very much for your presentation. It was really good. Okay, then the next candidate is Martino Alfredo Cappelluti. Hello. Again, the cure term is basic and applied immunology and oncology, and the title of the presentation is Development of an Epigenetic Editing Strategy Targeting PCSK9 as a Therapeutic Option for Hypercholesterolemia. So we just switch gears here and we talk about something that's not a tumor. Also,
I will try to change the topics a bit.
Yeah.
So, as you already said, the main topics of my thesis was to develop a clinically significant strategy to silence PCSK9 as a possible therapeutic approach for hypercholesterolemia. This is just a brief recapitulation of my presentation. I will start with a brief background. Hypercholesterolemia is a quite common condition characterized by abnormally elevated level of circulating cholesterol, the so-called LDL cholesterol. The main clinical outcomes remain the occurrence of coronary diseases. A class of really heterogeneous diseases includes stable events such as stable angina, but also acute and fatal events such as myocardial infarction. Just to give you the dimension of this problem, almost 50% of the death in Europe depends on CHTs, and this has an enormous consequence from an economic point of view. Almost 55% of the total health expenditure depends on CHT management. The central event in this diseases remain the atherosclerosis, so the formation, the inflammation, and the calcification of the plaque. Many cases have been stratified to be the cases of hypercholesterolemia, and this includes the life risk factors such as inappropriate lifestyle, tobacco, or alcohol consumption, but also some mutation affecting cholesterol-regulated genes define a particular set of hypercholesterolemia called familial cholesterolemia. Among these genes, one gene that is particularly attracting attention is PCSK9. The main function of PCSK9 is actually to regulate the expression of the LDL receptor cholesterols. These receptors normally bind the LDL, circulating LDL, and drive it to degradation. What commonly happens in patients affected by hypercholesterolemia, genetic one, is that gain-of-function mutation of PCSK9 determine an accelerated clearance of the receptors and so an accumulation of the LDL cholesterol. Different kinds of drugs have been invented to somehow limit the activity of PCSK9, and this includes really effective monoclonal antibody and small interferencing RNA. Both these approaches are crashing the clinics quite fastly, but it's also true that they require continuously and lifelong administration. To overcome this tedious lifelong administration, different innovative approaches have been invented more recently, and these include the genetic inactivation of PCSK9 by targeting specific artificial nucleases to the coded sequence of the gene. This might include Cas9, the so-fashioned Cas9, but also TALE and zinc finger. Upon induction of double-strand break in the coding sequence, what commonly happens is that the repair machinery will introduce random insertion and deletion that will determine the loss of gene function. This kind of approach has been already applied to PCSK9 and in vivo. As you can see, single administration of Cas9, mRNA expressing for Cas9 and single gather RNA targeting the first exon can really strongly inhibit the presence of PCSK9, almost cleaving PCSK9 from circulation, and this results in a really clinically meaningful reduction of cholesterol in the blood. The main limitation of this kind of approach remains the uncontrolled target activity of this platform. I just want to show you some of the possible outcomes that are not expected. So, for example, we are expecting that sometimes the DNA repair machinery may induce different outcomes, not just conventional insertion and deletion, but also it is conceivable that Cas9 may cleave to a different genome target from that one is expected to Cas. And more recently, it's been shown that administration of Cas9 and expression of nucleases in general and formation of double-strand break is not completely inert event. It may induce the activation of many sensors, including the P53 that finally will lead to cell cycle arrest and a more severe case also in the cell death. So this is something that we need to keep in mind when we think to extend nucleases activity for the clinics. To overcome this, let's say, genotoxicity event, our and other groups start to develop alternative strategies that do not require modification of the genome sequence. One of these strategies is the so-called engineered transcription repressors, accompanied by a conventional non-cutting DNA binding domain, for example, that one derived by Cas9, but dead Cas9, not retaining the activity of binding specific DNA sequence, but not introducing any kind of double-strand break. And this kind of domain typically is used to an effector domain, so a catalytic domain derived by natural enzymes such as epigenetic writer erasers. I just want to show you to list here two of this, the so-called de novo methyltransferases. This enzyme can catalyze the methylation of target DNA, and also and alternatively or in combination, this is conceivable to use the so-called recruiter domains, such as the CRAB, which main activity is just to recruit the endogenous transcription factors to do the side locus. This kind of approach has been really applied to different kinds of cell lines. Just I want to show you one of this. The transient expression of the tribal combination of 3A, 3L, and CRAB domain was sufficient to induce permanent and potent and time-stable repression of beta-2M in K562 cell line. So the idea of this project was to extend the application of this strategy to a clinically relevant gene such as PCSK9, and also in a clinically relevant and amenable to clinical translation setting. So what I did initially was just to start with a proof of concept in human and murine cell line. To avoid gene expression quantification, what I did was to generate two ad hoc cell line expressing PCSK9 in frame with TD domato reporter. So all the pictures that I'm going to show to you about expression of PCSK9 as just quantified as TD domato expression. Using this system where I initially performed the proof of concept in IPA16, a murine cell line derived by murine hepatoma, and I designed different single gather RNA targeting the CPG island of PCSK9, and I transiently expressed this single gather RNA in DTRs by plasmid transfection. As you can see by this plot, the expression of many of the single gather RNA, and in particular single gather number 4, was able to install a robust and time-stable repression of PCSK9. These results was really promising, but anyway, it was achieved by plasmid transfection. This is something that cannot be easily applied to in vivo, and for this reason, I optimized the platform as a possible fully mRNA platform containing mRNA that was chemically modified for it, and single gather RNA that was chemically modified. This kind of chemical modification allow longer expression and less immunogenic expression of TTR in vivo. For sake of time, I cannot show you data about this optimization, but I'm prone to answer any question you want to. Having optimized the platforms, then I start wondering if our platform can be really solve the off-target events that we were expecting to have in the Cas9 setting. To achieve this result, what I did was to combine information from a whole genome expression profile and whole genome methylation profile. What I did was actually to silence PCSK9 again in IPA16 to sort silent cells and then to quantify the expression of the whole genome expression of these cells. As a control, I used cell treated just with ETRs and not single gather RNA, or cell treated with conventional Cas9 and single gather RNA cutting the first exon. I'm not going to show you the plot here, but anyway, as negative control, I also had a mock treated cell. As you can see by this RNA sequence plot, and as expected, treatment with single gather RNA ETRs targeting PCSK9 produces significant and potent inhibition of PCSK9, but no other gene was found down or upregulated. And this was the same also for Cas9 treatment. This kind of results indicate that ETR treatment was not able to install any repression to transcriptional repression to other genes different from the intended one. This was not sufficient. We want also to profile the whole genome methylation of the treated cells. So we sorted again negative cells, and then we extract genome DNA and then analyze the methylation status by whole genome bisulfite sequence. As first, I just want to show you this CpG distribution in a, let's say, kilo-base window, 11 kilo-base window surrounding the promoter of PCSK9, so the target region. As you can see, treated cells do not present any methylation in the promoter region as expected from a highly expressed gene, but upon ETR transfection and single-guide RNA expression, this regulation gets, this region gets highly methylated. This was not true as expected for Cas9 treated cells. We are actually still doing whole genome analysis. I just want to show you some preliminary results. What we did was actually to subdivide the entire genome in 2.5 kb long beans randomly assigned, and to any of these beans we calculate average methylations. And what I want to show you is the difference between average methylation in mock treated samples and ETR, so Cas9 treated samples. In the case of ETRs, any red dots represent different methylated beans. In this case, we are dealing with hypermethylated beans. In this case, it's with hypomethylated beans. We found some differently methylated beans with a relatively low percentage. What actually was even encourages was that the same almost the same percentage was even found in the Cas9 treated cells, indicating that most of this activity might be more dependent on cell treatment per cell. So, for example, cell transfection, cell sorting, and so on, that more than ETR activity, since, as you know, Cas9 do not contain any methylation activity domain. So, having proven that our platform is effective and still safe, we start wondering in which way we will be able to deliver in vivo. And as actually vaccination is all teaching us, one of the most effective ways to deliver mRNA in the liver, but not also in the liver, should be the using of so-called LNP. Just to give you a brief idea, as LNP are formed, the main elements remain so-called cationic lipids. A positively charged cationic lipids is able to strictly interact and package up negatively charged RNA. Different kind of additional molecules have been implemented to stabilize and to help the delivery of this platform. This includes phospholipids, cholesterols, and PAG. Actually, we did our experiment using this property formulation system. The company is called Precision Nanosystem. They just use a microfluidic devices that can high pressurely mix RNP and cationic lipids, forming highly efficient and stable LNP. What I initially did was to test our this kind of platforms by conventional gene editing, so just delivering of mRNA of Cas9 as a positive control. And as a sort of surrogate readout. As a first step, I tested in cell line, as you can see by this plot, and what I retrieved was a high efficient knockout efficiency of PCSK9, even at relatively low doses, such as 60 nanograms. In parallel, we also performed an in vivo experiment injecting different doses and different peaks of LNP. And as you can see, a relatively low dose injection, such as 1.5 milligram per kilo, was sufficient to induce more than 60% reduction of circulating PCSK9. These results was actually speculated also by genetic analysis, where you can see that almost 50% of the gene has been edited by Cas9 delivery. Having found that this LNP can efficiently deliver Cas9 to the cells and in vivo, we start wondering if the delivery of ETRs by LNP could be as efficient as Cas9. And we did a similar experiment delivering ETRs in single-guide RNA targeting the CpG island. As you can see by this plot, we were not expecting actually that our platform always seems to be less effective than Cas9. At this point, we start wondering if this limitation might depend on the delivery vehicles, and so we performed a set of different transfection experiments using different methods, not just LNP, but also conventional liposuction and nucleosuction. And we need to say that any of the tested methods, Cas9 outperform ETRs. What was something that was quite fine to find was that at optimal transfection condition, anyway, ETRs can at least recover efficiency of Cas9. So we still need to optimize this step to arrive at the same efficiency. And actually, was what I did. I performed a dozen of a round of optimization of our platforms. I just wanted to show you two of the most effective. The first one was to change the ratio among the ETRs, the weight-weight ratio among the ETRs and the single-guide RNA. Just to give you an idea, the use of 1:1 to 1:4 ratio induced a twofold change in the activity of our platform. And this might indicate that despite we introduced a bunch of chemical modification within the single-guide RNA and the mRNA of ETRs, we still need to optimize and to stabilize the single-guide RNA. In parallel, I test in vivo further formulation, new formulation, again from precision nanosystem, again using Cas9 delivery as surrogate readout. And we identify some of this formulation that might outperform formulation number 2, so the formulation that we initially used. Using this formulation that so far seems to be the most effective, I decided to prove the efficacy of ETRs in a more clinically relevant, biologically relevant system. So primary hepatocyte derived by 2 different strains of mice, C57 and CD1 strain. And I did this analysis by transfecting both ETRs and Cas9 and measuring the expression of PCSK9 both at protein level and transcriptional level. As you can see in both the strain and respectively by the maturation of protein or mRNA, what we found was that the ETRs as Cas9 can efficiently reduce the expression of PCSK9. So this actually drives me to the conclusion. What I was able to show to you was that we can reach a really efficient long-term stable repression of PCSK9 in both human cell line and murine cell line. For sake of time, I did not show you some data about human, but we have completely similar results. I was also able to develop a fully modified and fully mRNA platform, so compatible with in vivo administration. Also, I identified many different platforms that would be useful for non-viral in vivo delivery of mRNA. And I also show you that this vehicle is really effective. We can reach more than 90% inactivation of PCSK9 when delivering Cas9. Also, I used to identify multiple paths to further increase the dose effective of our platform. I'm still doing it because we still need to refine and optimize our platform, but so far we need up to 8-fold increase in respective with the initial platform. I also was quite fine to discover that our platform can induce 80% reduction of PCSK9 in the primary cell line, so in the primary cell model, so really irrelevant. What I'm doing right now is I'm still further analyzing the whole genome by sulfide sequencing. As I say, it's quite a complex analysis. Maybe we may need to introduce a new kind of analysis. It might be more whole genome, but even with a higher number of quantification, let me say. In case we identify some potential of target, we are already thinking to multiple strategies to reduce the target activity of our platforms. Also, I'm still working on ETR dose effectiveness. One way should be to further characterize this new formulation that seems to work better than initial formulations. And I'm finally try to change the molecular architectures of ETRs that we believe is the case of lower dose effectiveness when compared to Cas9 activity. To do so, we are actually trying to change different linkers among different domains. We are changing DNA binding domain. Also, we are thinking to fine-tuning the effector domain. I hope that soon we will be able to modify, to implement all these modifications in the in vivo silencing experiment. I also did a second part of the project in which I try to expand the ETR's activity to a second clinically relevant target. This is actually HPV. For the same reason, for sake of time, I had no time to show you some data about it. But if you want to ask me a question, it would be a pleasure to answer to your question. I just want to thank all the people in the lab because they are actually helping me a lot. In particular, I want to thank Valeria. She's really working a lot in vivo part. All the data that I showed that was performed in vivo, they just rely on Valeria work. So I need to thank a lot Valeria. Also, I want to thank all the people that from other lab who are helped me a lot in performing experiments. Sarah is our bioinformatician, working a lot on whole genome and RNA sequence data. I also want to thank people that actually, I did not have the time to show data on HPV, but they helped me a lot. They still teach me how to work with the viruses, so in different kinds of biosafety levels. So it's something that really helped me a lot. And all the people that follow me during the PhD, during the commission, and all the university, and all of you for your attention.
Thank you very much for this very well organized presentation. Good. Thanks a lot. Any questions?
I try to find some connection with cancer, but so given the name of the program. But anyhow, no. So at this point, is I would say maybe technical development of maybe what can be within in the liver metastasis actually.
Sure.
So my question is referring to the nanoparticles that you used. So you mentioned differences in formulation. What is the difference between different formulation? Because at the end, to target the liver is something easy, that everything is going to the liver and it's now for nothing. So I just wanted to understand if there is some active targeting or just passive targeting.
Okay, in general, it's quite true that everything goes to the liver, but we just don't need just to go to the liver, but to specifically target the hepatocyte. There is something that could be not very easy because of non-hepatic cells still represent a big percentage of the liver per se. And to help the targeting of the hepatocyte, different kind of approaches being implemented. Many of the implemented lipid components seems to be quite specific for LDL receptor per se. LDL receptors is really expressed in the hepatocyte, and for this reason, this formulation seems to work really good targeting the hepatocyte more than macrophages, for example. Second, if I would still to answer to your question, it's true that formulation keeps, let's say, a conserved backbone, but it's also true that different components can be tuned. This is the case for helper lipids. For example, that a bunch of different helper lipids exist, and many of that may help both targeting specific organs and enhance the efficiency of targeting. In the case of tumors, for example, we also are thinking about this hypothesis. We are running some preliminary experiments. We're planning to run some preliminary experiments to measure the activity of this platform of LNP to targets and to enter in a tumor mass. But we still need to do some experiments to properly understand this kind of hypothesis.
Just practical. Do you have a real stake in the formulation of the nanoparticles?
No, it's just a matter of company. We don't have any information. We just have, let's say, a general information about the macromolecular structures, because they say that it's a conventional structure based on this function. So we just have some general line, let's say, but we do not have the property information.
I have a question on the introduction. Probably I missed it, but what is the function of PCSK9 and which kind of protein is? Because
maybe I went too fast on that point.
Yes, I think so.
I
missed it completely. So at the end, I don't know what the protein is.
Okay.
I think it's quite important.
Yes. PCSK9 was really, let's say, fashioned as a possible target because of its activity on LDL receptors. It's a sort of false protease. This is the way they used to call it, because it is described as a protease, but during protein maturation, it lost its activity of protease because the prodomain remained attached to the catalytic task, and this blocked the activity. The main function, as Too Fast said before, was that the PCSK9 bind LDL receptor as the LDL cholesterol binds the LDL receptor, but differently from what happened when LDL cholesterol bind the receptors, a PCSK9 chaperone LDL receptor to the degradation. That's why overexpression or gain of function mutation in PCSK9 induce an extra physiological degradation of the receptors and thus accumulation of LDL cholesterol.
Yes.
Do you see or do you expect any toxicity in the hepatocytes or?
Yes, we see. We measure some toxicity. I have to say that was really a minor toxicity, and it was more dependent on the formulation because we were able to measure the same toxicity when we deliver empty LNP, so just the vehicle. And we also need to say that this toxicity came out that dosage that is not compatible also with any in vivo administration. It was just a try. So as a general consideration, LNP seems to be quite well tolerated, let me say. If we do some kind of parallel with AV injections, that so far remained gold standard for the target liver, we assist with similar, even lower toxicity. So toxicity seems to be not to be a major issue.
It maybe comes from the
yes, from the chemical composition, purification, and stuff.
Any other questions? No. Okay then, thank you very much. We can move on to the next candidate, Valentina Pieri. The curriculum is Neuroscience and Experimental Neurology. The title is Advanced Diffusion MRI to Unravel Structural and Microstructural Constraints for Mini-Invasive Neurosurgery, from the ovine model to the human brain. I was interested in the costs of animal housing for ovine models.
But they said it. At the end, they said it.
No, unfortunately, in the end, they are being sacrificed.
Okay.
Okay, good morning to everyone, and thank you for being here. Today, I briefly talk about what I did during these 3 years. And as anticipated, I conducted experiments both on ovines as well as on humans. Our unit, the functional neuroradiology unit, was part of a consortium of institutions participating to an Horizon 2020 project called AIDEN 2020, Enhanced Delivery Ecosystem for Neurosurgery in 2020, that is based on the, is there a mouse?
I have a mouse.
No, it's okay.
No, the mouse is there, but probably you have to select the pointer.
If you go to the bottom left corner, you select the laser pointer. That's perfect.
It's based on the invention of a new prototype of flexible catheter by the engineers of Imperial College of London that is capable of following curvilinear trajectories within the brain, thus avoiding critical obstacles. Of course, in order to plan the insertion of this catheter, advanced neuroimaging such as diffusion MRI should be inserted in this automated system, because in the end, the catheter is inserted robotically. So in this context, my PhD project was to aimed at performing cutting-edge neuroimaging in living sheep, that was our model, paving the way potentially toward its clinical translatability to human patients. So the main goal was to integrate the AIDEN 2020 catheter and robotic delivery with advanced diffusion MRI and experimentally test it. So first of all, by defining the microstructural properties of the ovine brain tissue and building an imaging tractography atlas. Then testing the system in vivo in sheep, performing convection enhanced delivery of a gadolinium solution. Then by translating the applicability of the platform to humans, simulating, planning for deep brain stimulation, and in the end, by increasing the specificity of the preoperative planning by enhancing an algorithm for quantitative tractography. So the experiments I will present were conducted in sheep and in humans. This is the outline of my presentation, and I hope the blue bar at the bottom of the slide can help you following what I'm presenting. First of all, starting with the project and the basis of diffusion MRI, I just want to point out that a flexible catheter may have potentially many microsurgical applications in the brain, but the two most important ones are convection enhanced delivery, so the local release of therapeutic directly in the brain, and the deep brain stimulation, so the electrical impulses delivered directly in deep brain structures. For both those applications, a flexible catheter may enhance the current treatment and avoid possibly critical anatomical obstacles, which is fundamental within the brain. In this context, the integration of diffusion MRI within the system may help in depicting more specifically microstructural tissue features that are not visible with standard MRI, and also depict obstacles that cannot be detected in any other way. As far as diffusion MRI is concerned, this is a very it's a peculiar interest of the functional neuroradiology unit of San Raffaele, and it's a non-invasive method to characterize in vivo the tissue microstructures. In order to interpret the raw data of diffusion MRI, there are many methods, and in particular during my PhD, I mainly focused on diffusion tensor imaging and NODD, so new right orientation, dispersion, and density imaging. Just to mention them, DTI is a model-free method to quantify the diffusion, and it can distinguish isotropic diffusion in a gray matter, for example, where the diffusion is identical in all directions, and anisotropic diffusion, for example, within the white matter, where the diffusion of water molecules follows preferentially the direction of axonal bundles. NODD, on the other hand, is a complex and sophisticated model that goes beyond DTI and also differentiates within brain tissue three different compartments: the intraneurite diffusion, the extraneurite diffusion when water molecules are hindered by the extracellular space, and the free water diffusion, so in the CSF. So given those bases, first of all, in the context of Eden 2020, we want to study our model because there were not many DTI studies on sheep. And as a first interesting thing, the sheep brain is very similar to the human brain as far as the vasculature is concerned, and it's a gyrencephalic model. So it has gyri and sulci, and functional motor areas that can be recognized. So our aim was to characterize deep its structures by optimizing a protocol, performing MRI tractography in sheep, and integrating white matter tracts in a standard atlas. So we performed acquisitions of images in 10 healthy sheep and developed the deterministic tractography, and then superimposed the tractography on T1 anatomical images. So this is a very snapshot of an example of the 5 white matter tracts that we managed to reconstruct in all the 10 sheep: corticospinal, corpus callosum, the fornix, frontoccipital fasciculus, and the optic radiations. And since our results were very reproducible between sheep, we actually overlaid the same tract of all the 10 sheep and represent it as a unique tract, fiber bundles, that can be visualized in a color-coded way, so that in red voxels you can see portions of the tract that are common to at least 4 sheep out of 10, while in yellow voxels it's visible the most consistent portion of the tract that's identical between all the 10 sheep. So in the end, we built the first MRI tractography atlas of sheep, and it's the first demonstration of a comprehensive characterization of tissue in the ovine brain. Our results were very reproducible and can be very useful in the future for standardized imaging analysis, because the ovine model is quite frequently used for neuroscience research. Then we moved on. After having characterized the sheep, we could use it as a model for in vivo experiment, and we infused a gadolinium solution with the aim of characterizing drug distribution after convection-enhanced delivery. So besides assessing the impact of brain microstructure per se on the infusion, we also performed 2 different kinds of infusion, parallel or orthogonal to white matter fibers, and assessed if any difference were present. So in this experiment, we explored the approach of convection-enhanced delivery. 7 sheep received bilateral infusion for a total of 14 infusions. So as you can see here, after reconstructing the corticospinal tract, it was reporting on T1 images. An entry point and a target point were selected for inserting the catheter, and then infusion was performed with a micropump at 3 microliter per minute, so very slowly. After analyzing all those data, what was clear that the drug distribution followed actually the white matter distribution. So we assessed a greater drug distribution along the main direction of white matter. Of course, it could be thought that it was predictable, but instead it has never been demonstrated before in vivo. Moreover, by infusing the drug or parallel to the fibers or orthogonal to the fibers, we assessed it after stopping the CAD procedure that the shape of the bolus infused parallel to white matter fibers was more oval than the shape of the bolus infused after orthogonal infusions, meaning that also the catheter positions may play a role. So this is very important because the CIG trials are not very successful until now, and so helping in predicting the final drug distributions depending on many variables can be useful also for future studies. Moving to human subjects, we actually wanted to test if the integrated system could work also with human images, and we wanted to integrate MR tractography within the planning algorithm simulator. In order to compare it with the standard of care, we wanted to test curvilinear trajectories with rectilinear trajectories that are the ones that are used now in the clinic. So here we tested the deep brain stimulation approach on 10 healthy subjects, and in those we identified also the corticospinal tract as a critical obstacle to be avoided by the planning algorithm. So we selected in the middle frontal gyrus 10 different entry points, and then we selected as the target of the stimulation the subthalamic nucleus that the actually target used in the clinic, and from each point to the same target point, different rectilinear and curvilinear trajectories were computed by the algorithm. And in the end, we assessed that after telling the algorithm to avoid critical structures as the best as it could, if it computed rectilinear trajectories, anyway, it was very near to the obstacles, while computing curvilinear trajectories, the catheter can find a better balance and avoiding critical obstacles. And here is an example of the same target point with a curvilinear trajectories that better preserves the globus pallidus here. So in the end, in our computational model, we demonstrated that the integration of MR tractography is feasible also for the human patients, and advanced imaging can be integrated in a preclinical evaluation of those kind of surgical procedure, and then also that our planner can automatically compute trajectories and curvilinear trajectories, ensure more safety with respect to the rectilinear ones. Finally, my final work was also a very interesting and technical part on the tractography analysis, was to deepen the standard tractography analysis and also quantify the diffusion metrics along the course of white matter tract by optimizing a new method, a new algorithm in MATLAB in order to extract those values along the tract. We firstly wanted to build a reference standard database of healthy controls because it has never been published before, so we didn't have a reference value. And then we analyzed the patients with brain tumor, so pathological conditions, and assessed if differences could be seen between the healthy and the patients. So we analyzed the 15 healthy controls and 22 patients, performed tractography in all of them, and then applied our new algorithm. So the tractography, just to show you how it is represented in humans, here is an example of the four very important eloquent tracts in the human brain: the arcuate, ascendens, inferior frontooccipital fasciculus, and the uncinate. So in all of the fascicles reconstructed for both controls and patients, we applied our algorithm that reparametrizes the streamlines to match correspondences between points that belong to the same tract cross-section. So all those points are collapsed in a single point, and a kind of skeleton of the fiber tract is obtained. Skeleton through which, point by point, in particular along 100 points, quantitative values are obtained. So our first result for all the tracts analyzed, this is our reference standard database of the 15 healthy controls, showed that actually the intraneuritic diffusion that I showed you before, extracted by Noddy, was always the most representative component along white matter tract, as expected, while the extraneurite component, so the extracellular space, is reduced, and in healthy tracts, free water is almost zero. The yellow line represents the orientation dispersion that varies along the course of the tract. Our most interesting finding was that when we compared patients with pathologically infiltrated tracts that were infiltrated by tumors, brain tumors, actually the metrics deviate of more than 2 standard deviations from the reference standard. And this is interesting because just reconstructing tractography, the tract can appear healthy, otherwise if they are completely destroyed, because the tractography is an algorithm that estimates the direction of fibers. So even if some portion of the fibers is partly damaged, sometimes the algorithm can cope with it. While analyzing in detail, voxel by voxel, the quantification of the metrics, subtle differences can emerge. And this is very important because the greater accuracy of Noddy in quantifying diffusion features along the tract can help in highlighting pathological tract profiles, and so possibly guiding also the surgical approach. And providing a quantitative signature of microstructural changes of peritumoral white matter. So just to summarize, our aim was to integrate the platform with MRI and test it. And we actually, in sheep, we firstly studied the model, building an atlas, and then used it as a in vivo model for experiments in vivo for CAD. And then in humans, we computed algorithm for DBS procedures and expanded the field of quantitative tractography. I want to thank all the people that allowed me to work in this very interesting and multidisciplinary project, Professor Falini and Dr. Antonella Castellano, also Rossella Galli, because I collaborated also with her lab, and Professor Massimo Filippi, because now I'm working in this unit because I entered the residency program here.
Wow. Thank you. So, could you just summarize, give me a brief sample of potential applications? For instance, you talked about targeting the subthalamic nucleus, and that I suppose has to do with Parkinson's disease.
Yes.
So, how do you envisage doing that? What type of, what will you deliver into the subthalamic nucleus?
Okay. Well, nowadays the subthalamic nucleus is stimulated with electrical impulses, but for example, also in this field, one of the main limitations is that with the rectilinear trajectories, only the most anterior portion of the nucleus can be reached, and so sometimes the therapy is not completely effective, or at the end, the patient can develop resistance to this therapy. And one of the aim of a curvilinear approach is to better reach the subthalamic nucleus, possibly stimulating a more enlarged area. Moreover,
does the stimulation have a permanent effect?
No, it should be constant.
So, how invasive is this approach in a patient with Parkinson's?
It's quite invasive. In fact, it's a neurosurgical procedure, and then the patient is in follow-up with imaging and electrical control in order to recalibrate the stimulation depending on how the clinic works. So another aspect is that with turbinary trajectories, since the subthalamic nucleus is surrounded by very critical structures, sometimes the patient can bleed during the procedure, or some other structure can be damaged. So
internal capsule fibers of all types. Yeah.
Instead, for the delivery of drugs, is more envisioned with respect to tumors, even if actually one of the problems is that also therapies are missing, but just starting to deliver something that actually enters the tumors and not goes around would be a good start.
Yeah, we've heard some potential applications in it that approach to glioblastoma, for instance, and there are some that are based on immunotherapy, others that are based on gene therapy and modulation of it. So this would be very nice. It would probably take care of one of the basic problems that is delivery, accurate delivery to the target organ. That's nice, very, very interesting. Any other questions?
Maybe I missed it. So everything you showed is invasive. Or
the
last part.
The application of the catheter is invasive because it must be inserted inside the brain, but the tractography imaging is not invasive.
So that can be obtained in a regular MRI?
Yes, it is actually currently obtained in the clinic. In fact, now for preoperative planning of...
So you really see the diffusion along the different...
Yes.
But in the end, subjects, you didn't use the catheter, I mean humans, or did you? Because you presented...
No, in humans, we didn't use the catheter yet, yes.
Right, no, sorry.
No, go ahead.
No, it's just curiosity. I don't know where the sheep were kept.
No, it's the University of Lodi. Has a veterinary center very big, and they order the sheep in pairs every month because they feel lonely if they are if they stay alone. And so we organized that we went there once every month and performed the experiment. Yes.
They're not sent to. No, but this is part of it.
No, yes, it's an horizon project.
Did you perform yourself?
Yes, well, we went personally to Lodi every time, and I didn't do anything on the ship because there were veterinary anesthesiologists and neurosurgeons. I performed the brain MRI acquisitions on the ship because Dr. Castellano optimized the protocol for diffusion acquisition, and I studied comparative anatomy of the human brain and the sheep brain together with the veterinary team, and slowly we started to find where the fibers can be reconstructed also with MRI.
So I assume there are no metal parts in the catheter, of course, because it has to be...
No.
What is the material?
It's actually there is a guiding catheter that's plastic or something that after the insertion of the silica catheter that's in glass is removed. So what remains inside is a silica catheter for the infusion.
Thank you very much.
Thank you.
Okay, the next presentation, again for the curriculum of neuroscience and experimental neurology. It's about a very fascinating project carried out in the Muzio lab. It's presented by Riccardo Sirtori, and the title is Therapeutic Effects of Retromer Stabilization in Amyotrophic Lateral Sclerosis.
Okay. My name is Riccardo Firtori, and today we'll talk about my PhD project, Therapeutic Effects of Retromer Stabilization in Amyotrophic Lateral Sclerosis. Retromer is a multi-coating complex whose function is to relocate endosomal cargoes from the endosome to the transgolgi network of the plasma membrane. The retromer complex is formed by two great subunits, the cargo recognition core and the recruitment module. The cargo recognition core is formed by VPS26, VPS29, and VPS35. The last one is the central element of the cargo recognition core and is fundamental to interact with the cargo protein. The recruitment module is formed by SNX proteins, which are located on the endosomal membrane and are fundamental to recruit the cargo recognition core on the endosomal membrane. From 2005, increasing evidence has been collected about the role of the retromer complex in different neurodegenerative disorders. In particular, low levels of VPS35 and VPS26 were observed in postmortem biopsies of the dentated gyrus of patients affected by Alzheimer's disease. In 2011, was described for the first time the mutation D620N on VPS35, which is causative of an autosomal dominant form of Parkinson's disease with late onset. In 2019, low levels of all the members of the cargo recognition core were observed in postmortem samples of the frontal cortex of patients affected by progressive supranuclear palsy and Pick's disease, which are two different tauopathies. But the most important for us, in 2017, low levels of VPS26 were observed in iPS-derived motor neurons from ALS patients carrying C9orf72 mutations. So my group decided to study the role of the retromer complex in amyotrophic lateral sclerosis, a severe neurodegenerative disorder characterized by the loss of the lower and the upper motor neurons, causing muscle weakness and hyperreflexia. We approached the study of the retromer complex starting with a well-known Mri model of ALS, which is the SOD1 gene mouse model. These are transgenic mice which carry moderate to epic copies of the human SOD1 gene. The clinical onset of these mice is near day 90, so we decided to study the retromer complex of these mice at two preclinical time points and one near to the clinical onset. As you can see by immunofluorescence, we observed a reduction of VPS35 and VPS26 levels in positive neurons in the ventral horn of the lumbar spinal cord of gene-edited mice compared with wild-type controls at all of these three time points. We further confirmed the reduction of VPS35 levels by Western blot on lumbar spinal cord extracts from gene-edited mice at day 20, day 60, and at day 100. We also observed a reduction of VPS26B, which is a particular isoform of VPS26 highly expressed in motor neurons, and VPS29 in the spinal cord of gene-edited mice at day 100. Interestingly, we didn't observe any significant difference in the gene expression of all the members of the cargo recognition core in the spinal cord of gene-edited mice compared with wild-type mice at day 30, day 60, and at day 90, meaning that retromer impairment is probably a post-transcriptional event. Basing on these results, my group decided to use the retromer complex as a therapeutic target. Our collaborator, Professor Pierfausto Seneci from Università Statale di Milano, developed a new class of compounds starting from R55, the first molecule described to interact with the retromer complex at the interface between VPS35 and VPS29. Professor Mario Milani screened these compounds, studying the interaction between these different compounds and the retromer complex, and evaluating the KD of this interaction. Then we screened the selected compounds basing on their ability to increase VPS35 levels in vitro. So we treated neuro2A cells with these compounds using R55 as our positive reference. And we selected compound 2A as our leading compound, since it acted better than R55 to increase VPS35 levels in vitro. It has the best KD of interaction and was the most lipophilic molecule. We also observed that compound 2A is able to increase not only VPS35, but also VPS26 and VPS29 levels in vitro. Basing on these results, we decided to move in vivo. At first, we had to demonstrate that compound 2A, as predicted by Professor Seneci, is able to cross the blood-brain barrier. To do so, we performed a sort of PK assay, treating wild-type mice with compound 2A, injecting them intraperitoneally every day at a dose of 10 mg per kilo. Then we sacrificed these mice at different time points, and PROMIFAC technicians performed mass spectroscopy. They observed a detectable amount of compound 2A in the brain of these mice starting from the very first time point, and they also observed that the amount of compound 2A increased during the time. We also evaluated VPS35 levels in the spinal cord of wild-type mice treated for 1 week, and we observed that compound 2A, as we expected, was able to increase VPS35 levels in vivo. Basing on these results, we decided to move in ALS pathologic field. So we treated SOD1 genetic 3A mice with our compound 2A, starting from day 30 to day 100, injecting them intraperitoneally every day at a dose of 10 mg per kilo. Starting from day 50, we tested the motor performance of these mice using the rotarod assay, which measures the last Tesi to fall time of the animal on this machine. And as you can see by this graph, we observed that genetic 3 mice with compound 2A performed better than genetic 3 mice treated with vehicle, and this difference was statistically significant starting from day 90. In order to understand how can compound 2A improve the motor performance of our mice, we decided to evaluate the number of survived motor neurons in the ventral horn of the lumbar spinal cord of these mice using 2 different techniques. We evaluated NEON positive motor neurons, basing also on the morphologic criteria, since NEON is not specific for motor neurons, and we evaluated child positive motor neurons. With both these 2 different methods, we observed a higher amount of motor neurons per section in the ventral horn of the lumbar spinal cord of genetic 3 mice treated with compound 2A compared with genetic 3 mice treated with vehicle. The last step was to understand the mechanism of action of compound 2A. At first, we had to demonstrate that compound 2A is able to ameliorate the retromer deficit we observed in genetic 3 mice. We did so by immunofluorescence and Western blot, and we observed that VPS35 and VPS26 levels were higher in the spinal cord of genetic 3 mice treated with compound 2A compared with genetic 3 mice treated with vehicle. Next step, we decided to study cation-independent monophosphate receptor, since CIMPR is one of the most important cargo of the retromer complex, whose function is to deliver hydrolytic enzymes to the lysosomes, and lysosomal impairment is a common feature of neurodegenerative disorder. As you can see, we observed a reduction of CIMPR levels in NEON-positive neurons in the ventral horn of the lumbar spinal cord of genetic 3 mice treated with vehicle compared with wild-type mice. But also in this case, we observed that compound 2A was able to partially restore CIMPR levels. Given that compound 2A is able to ameliorate CIMPR levels, we decided to study the delivery of hydrolytic enzymes to the lysosome. So we studied cathepsin D. Cathepsin D is an hydrolytic enzyme which has a maturation process during its voyage from the trans-Golgi network to the lysosome, with a progressive reduction of its molecular weight. So the ratio between light chain cathepsin D and heavy chain cathepsin D is a good tool to evaluate the delivery of cathepsin D to the lysosome. And as we expected, we observed an higher ratio between light chain and heavy chain cathepsin D in the spinal cord of gene-engineered mice treated with compound 2A, meaning that compound 2A was able to improve cathepsin D delivery to the lysosome. The last step, as we expected, improving cathepsin D delivery to the lysosome and lysosomal activity, compound 2A is able to improve SOD1 toxic aggregates. In order to demonstrate this idea, we performed a blue native gel, which is a Western blot in not denaturative and not reducing conditions. And we observed a reduction in the total amount of SOD1 toxic aggregates in the spinal cord of gene-trained mice treated with compound 2A compared with gene-trained mice treated with VECO, meaning that compound 2A is able to foster the clearance of SOD1 toxic aggregates. So, briefly resuming the most important results, we demonstrated that retromer is impaired in SOD1 gene-expressing mice. We demonstrated that the novel class of compounds designed by Professor Seneci is able to increase VPS35, VPS29, and VPS26 levels in vitro. We demonstrated that compound 2A is able to cross the blood-brain barrier and to increase VPS35 levels in vivo. Administering compound 2A to SOD1 gene-expressing mice, we were able to ameliorate the motor performance of these mice and to reduce the motor neuronal loss. Probably the mechanism of action of compound 2A relies on the increasing of CIMPR levels, fostering cathepsin D delivery to the lysosome. In this way, compound 2A is able to ameliorate the motor neuron capability to degrade SOD1 toxic aggregates. Special thanks to my docs, Luca Muzio and Milo Riva, who were the head of this project, and the technician Andrea Fossaghi. And thanks to all the members of the laboratory of Professor Gianvito Martino. And thank you all for the audience.
Very nice.
I have a question, if I...
Yeah, sure.
So at the beginning, you were saying, if I understood correctly, that this protein that you studied was mutated, was found that there is a mutation.
No. In ELS, no.
Okay. It's only in Parkinson.
Yeah.
Okay. Because I was wondering what was the rationale to go and check in the mutant mice of SOD this specific protein.
In different neurodegenerative disorders, there is an impairment of protein trafficking, which leads to an impairment of different systems related to protein trafficking. So we decided to study this system, which was found impaired in different other neurodegenerative disorders.
So, is there an effect on the lifespan, on survival of these SOD1 G93A mice?
We don't know. We didn't check the survival of these mice.
And why was that?
Because the most important outcome is the motor function, because in different studies relies on the survival of the mice survival didn't prove a compound able to go to the clinic.
On the other hand, the maintenance of motor neuron positive for what marker you used from a neuron in the ventral horn doesn't necessarily, I mean, the positivity for mu n in the ventral horn doesn't necessarily tell you much about the axonal terminal and presynaptic. And then this is a dying back neuropathy where the earliest events are.
I didn't show you, but we also studied with the help of Nilo Riva and Angelo Quattrini the axonal fiber degeneration in the sciatic nerves of these mice. And we observed a reduction in the degenerating fiber density in the sciatic nerves of mice treated with compound 2A.
Okay,
so partially.
Did you look at markers, presynaptic markers at the motor end plate?
No.
Okay. And the other question is, G93A is the best characterized model in mice because of its very predictable and reproducible behavior. But in humans, it only accounts for less than 10% of the sporadic cases, and whereas 90% is due to accumulation, nuclear depletion, and cytoplasmic accumulation of TDP-43. Are you going to look at TDP-43-based models? There are some recently, a new model came up that has a mutation. It's a knock-in model, N390D, I believe it's called. And despite, although in this mouse, the levels of TDP-43 are native physiological levels, but the phenotype is very robust and reproducible.
No, we didn't study the retromer complex in TDP-43 mutated environment, since our previous model of TDP-43.
I know very well. It's a complete disaster. Complete disaster. Okay. All right.
Can
So,
I ask a more general question, because I'm not in the field at all? So you're proposing potentially your approach as therapeutic option in the future based on your results. Can you help me understand a bit the landscape of innovative approaches which are being developed for this, which is fatal, of course, so how your strategy compares with the others, and there are many other possible?
Yeah.
nothing working. I will summarize the answer.
No, this approach has something different and something which is similar. The idea to improve the degradation of SOD1 aggregates is an old idea. The difference of this approach is that it does not push on autophagy or proteasome, but I think it ameliorating lysosomal homeostasis. It can foster indirectly this process. So,
and no other approaches as far as you know, at the clinical levels which are being tested as possible therapeutic options, other mechanisms or
other mechanisms possible to ALS.
you know, I expect this was in your introduction, so because this is a pattern disease and there are no therapeutic options working when available, and you are studying a novel approach to be possibly brought into clinics. Are you aware of any other different strategies that is being studied?
Yeah.
That's very general.
Yeah, there are a lot of strategies. Also trying to target mRNA of mutated SOD1 or TDP-43 using ASO or whatever. But I think that neuroprotection using a system which improves toxic species degradation can be used to a wide plethora of different ALS subtypes. So it's a more general approach.
Any other questions? Thank you very much. Working this way. Okay, The last candidate for today is Tiziana Tempio for the curriculum of cellular and molecular physiopathology, and the title of her presentation is A Molecular Machine That Controls Protein Traffic at the ER-Golgi Interface. It's spelled Golgi, but I'm confident that it's about the Golgi, not the Golgi.
Okay, so good morning, everyone. I'm Tiziana Tempio. It's a pleasure for me to be here and to see you.
Our pleasure too.
By person. So let's start with an important concept that concerns the ability of cells in maintaining organelle identity despite a continuous protein flux. Indeed, about 1/3 of the proteome is represented by protein destinated to the extracellular space, and they travel along the secretory pathway that is composed by the endoplasmic reticulum, the ER-Golgi intermediate compartment, and the Golgi compartment. And here is important to underline that only protein that are correctly folded can proceed to downstream compartment. And in this flux of protein is maintained thanks to a dynamic balance that exists in protein flux, depending on different transport mechanisms. Indeed, there are proteins that cannot exit from the ER for reason of size, while there are other proteins that instead need to move and to reach the downstream compartment. And we can talk about bulk flow mechanism when protein enter into the vesicle at the ER exit site by default. And we can talk about a cargo receptor mediated transport, referring to an active transport based on the recognition of selected protein by different cargo receptors. And in this scenario, it's important to underline the role of the retrograde transport because it is needed to maintain the correct volume and membrane composition of the different organelles. It's also important that this protein flux is very controlled because it's important to ensure secretome fidelity thanks to different quality control mechanisms. There is a primary ER quality control, a proximal checkpoint that occurs in the endoplasmic reticulum, and there is also a post-ER quality control checkpoint that occurs in the ERGIC and Golgi compartment. And here, active protein that is the focus of my PhD project, RP44. RP44 play a role in post-ER quality control with the help of other proteins. One is the lectin ERGIC53, and the other one is the receptor, the KDA receptor. RP44 is a dynamic protein that recycles continuously between the endoplasmic reticulum and the Golgi compartment and changes its conformational state depending on its localization. Indeed, in the ER, RP44 is in a close conformation with the active site that is masked by the tail, while in the Golgi compartment, RP44 is in an open conformation, the active site is exposed, is able to recognize the cargo protein, and moreover, it exposes the RDL sequence at the end of the tail that is needed to recognize the KDA receptor that is important to mediate the retrieval of RP44 back to the endoplasmic reticulum. This is the cycle of the protein, and that is important to control the secretion of some cargo protein and the localization of some cargo protein. And during my PhD, I try to dissect some parts of this molecular machine. And today, for reason of time, I will tell you about only the more relevant result. About, in the first part, the anterograde transport of RP44, and in the second one, about the RP44 activity and retrograde transport. So, the starting point correlates with the function of RP44. Indeed, being a retrieval protein, RP44 should be very efficient to reach its working place, that is the ERGIC and Golgi compartment. And this is because in order to overcome the cargo that need to be recovered, RP44 should move faster than this cargo protein. So the first question was, does RP44 move faster than other proteins? And in order to analyze the movement and the speed of RP44 into the secretory pathway, I decided to measure the secretion of the protein, comparing the secretion of a reporter with the same reporter fused with RP44 without the RDL sequence, so a secretable version of RP44. I try with different reporter in order to exclude some specific effect mediated by the reporter. But today I will tell you only about the allo reporter because it presents different advantages. Because allo is a molecule that specifically binds and irreversibly binds fluorescent allo ligand. And this is important because allow me to locate and track the movement of the protein of interest. And finally, but importantly, being a bacterial enzyme, allo should have no specific interaction in the endoplasmic reticulum, so it can be considered like a generic reporter that follows the bulk flow process. So I decided to take advantage of the allo properties, and I perform an allo visual purchase, comparing the secretion of the reporter allo with the same reporter fused with RP44 delta RDL. I transiently transfected the cell, and then I first saturated all the pre-existing molecules using an allo colorless ligand. And then I labeled the new synthesized protein in red using a red allo ligand. And in this way, I can follow the secretion only of the red protein that represents the new synthesized protein. And here we clearly see that at the beginning of the phase, the allo reporter in red does not colocalize with the Golgi marker GM130 in green, while on the contrary, allo fused with RP44 strongly colocalize with the Golgi marker. And after 2 hours of chase, while the reporter allo is reaching now the Golgi, some point in yellow start to appear, the allo RP44 left this compartment because after 2 hours it is secreted. And I decided to use the same approach, performing also a biochemical allo visual purchase. And in this case, I detect the fluorescent signal of the allo fluorescent ligand directly on the gel. And here we see that the intracellular material disappears, accumulating extracellularly for both the protein. But it's clear that the export of allo RP44 is favored here in red, compared with the export of the reporter alone, shown here in blue, demonstrating that RP44 is secreted more efficiently than a generic carboprotein. So the second question was, how can RP44 exit more rapidly than a reporter protein? And we focus our attention on the role of a cargo receptor, RGC53. And we hypothesize that RGC53 probably mediates an active transport for RP44, starting, of course, from previous data obtained in our lab. And yes, I demonstrated that the absence of RGC53 slowed down the export of allo RP44 that here we see accumulate intracellularly without any effect on the reporter allo. So the lectin RG53 acts as a transporter for RP44. This lectin exits from the ER using the interaction with the COP2 vesicle, and briefly, RG53 interacts with the internal layer of the COP2 vesicle that are formed at the ER exit site. And in particular, RG53 is able to bind the SEC24 component of this internal layer. And for this reason, I decided to use a drug, the 4-phenylbutyrate, that is a molecule that competes with the binding of RG53 to SEC24 and displays the RG53 to bind the vesicle. And I demonstrated that the use of PBA inhibits completely the secretion of allo RP44 that accumulates intracellularly, but with minor effect on the reporter. So demonstrating that the PBA selectively prevents the allo RP44 secretion, and in this condition, we can say that RP44 is selectively excluded from the COP2 vesicle. If we try to draw a model of this situation, RP44 and RG53 interact in the endoplasmic reticulum. They move together using the RG53 interaction with the COP2 vesicle, and they reach the Golgi compartment. And if we use the drug, the 4-PDA, we block the transport of RP44 because we block the interaction of the transporter with the vesicle. And this suggests that probably the two proteins stay blocked and accumulate in some region of the endoplasmic reticulum, with consequence, of course, on the RP44 activity. So affecting the RG53 activity, RP44 does not reach the Golgi compartment. The second part of my talk today regards the RP44 activity and retrograde transport and the role of the zinc. This because zinc, together with pH, control the RP44 tail opening. In the secretory pathway exist essentially two gradients, one gradient of pH and one gradient also of zinc. Indeed, there is an higher zinc concentration in the Golgi thanks to the presence of this zinc transporter that actively pumps zinc into the Golgi compartment. Here, RP44 is able to bind zinc, and this binding forces the open conformation of the protein. And we demonstrate that zinc can dynamically control the RP44 traffic and localization. And we demonstrate that using a zinc chelator, this molecule T10, that is an intracellular membrane permeable chelator, that removes zinc from the cell, RP44 stays in a close conformation and accumulates in the Golgi. And this is what we see in this image, that in which RP44 strongly colocalizes with the Golgi marker. But what is surprising is that this regulation is reversible, because the subsequent addition of zinc, of an excess of zinc, is sufficient to relocalize RP44 in the ergic and Golgi compartment as in the control, demonstrating that zinc controls the RP44 tail opening and the RP44 localization. And what about the RP44 activity? I demonstrated also that zinc is important and is needed to regulate and control the activity of RP44. One of the activity of RP44 is the retrieve of the some cargo protein. And I analyzed the secretion of two of these cargo proteins. One is IRAK1 and the second is IRO1 alpha. These two proteins are ER-resident proteins that lack the retention sequence to stay in the ER. So they rely on the activity of RP44 to stay in the endoplasmic reticulum. And I demonstrated that removing zinc, both using T10, so the zinc chelator, or specifically silencing the zinc transporters that are located in the Golgi, increase the secretion of both the two proteins compared to the control, demonstrating that zinc is needed for efficient and client retention by RP44. Of course, this effect is specific because PDI is never secreted. So if we try to put all together this information in a model, we see that in the endoplasmic reticulum, where the pH is higher and the zinc concentration is low, RP44 is in a close conformation. It can bind to RG53. They move together using the RG53 interaction with the vesicle, and they reach the Golgi. Here in this environment, the two proteins detach, and there is a decrease of the pH and a higher zinc concentration. And this environment is important because allow RP44 to assume its open conformation. In this condition, the active site is exposed, RP44 binds zinc, and expose the RDL at the end of the tail. And in this condition, RP44 can bind the cargo. This cargo protein can be a common cargo between RG53 and RP44, or also another cargo protein that reaches the Golgi independently from RG53. In this condition, RP44 is able to bind the KDR receptor and perform its function, its retrieval back to the endoplasmic reticulum. So the take-home message are that I demonstrated that RP44 exits the ER via active transport mediated by RG53, that zinc regulates the structure, the function, and the localization of RP44, and that the two proteins, RP44 and RP53, form a functional couple important in post-ER quality control. So I want to thank my director of study, Roberto Sitia, my second supervisor, Maurizio Molinari, and all the lab members, Tiziana, Chiara, Ilaria, all of these people that are really very important during this journey. And of course, all of you for your attention.
Beautiful presentation, lovely cell biology, and you already have a paper in press.
Yeah.
Where is it going to be published?
In a few, in science, in a few.
Oh, that's amazing, fantastic.
Thank
Great.
you. Thanks.
It's beautiful project. Wonderful. Any questions?
So you were measuring zinc binding to the protein. You're able to.
We personally okay, yes, we measure zinc binding, not I personally, but we collaborate with a Japanese group that measure the affinity of RP44 for zinc, that is sub-micromolar affinity. So we know for sure that RP44 can bind zinc because it presents a histidine region in particular between the last domain and the tail, and this is the site of zinc binding. And yes, and we obtain also in collaboration, of course, with this group in Japan, the crystal structure of RP44 binded to zinc, that is in a different conformation with respect to the close one. Yes.
So just one question, maybe I need to the detail. When you were showing that RP44 and RP53 were associated in order to be transferred, and then you were using the drug to interfere with their transfer. Do you have proof that the two remain assembled though they don't move to the histology, or is it complicated?
This is a very, very, very good question. Thank you. We have, first of all, we have, of course, the proof that the two proteins interact, also physically, from a co-immunoprecipitation assay or immunofluorescence assay, but we don't know in which is the domain of RP44 that specifically interact with RP53. We imagine that they interact in the ER for reason of transport, but we are not able, in condition of PDA, we don't see the secretion, but we are not able in immunofluorescence to understand exactly where they are located. Because I try to find they are associated in the ER, because I imagine that the RP44 cannot move, so stay in the ER blocked. And I try to check the co-localization with carreticulin, for example, and I don't find this localization. But there is an important point about this, because the ER is an organelle that probably is composed by like a gradient. There are some components that are more static, for example, carreticulin, and some other components that are more dynamics. So probably I checked with the wrong mark. I did not check with carreticulin, but I think that they are blocked in the ER in some region that probably is near to the ER exit site. We can try to use some other marker of the ER exit site and see if they are blocked and then co-localized in this station.
Any other questions? Okay. So may we, first of all, thank you very much again. It was a very nice morning of great science. And I really appreciate it. I'm sure I can talk on behalf of everybody else. I would like to ask you now to leave the room so we can discuss the presentations and then finally call you back inside for awarding the title.
Controlliamo che abbiamo interrotto. Ci sentono?
Tutto pronto?
È interrotto il collegamento YouTube?
Daniele?
Perché ci hanno detto di non toccare
No,
niente.
infatti, non tocchiamo. Sono addormentati.
Sentiamo un attimo, perché fare la discussione su YouTube non è buono.
No, non è bello.
Grazie. Il Presidente si reca un attimo in videoconferenza con il Giappone.
Va bene.
10 minuti finivamo.
Sto guardando.
Il primo nome su Science.
iScience è il primo nome, iScience.
Data communication, no, infatti, anche perché manca tempo stare dietro.
No, però si era tutto per farci vedere, perché si era tutto tutta la parte di struttura.
Su YouTube.
No. Parte di struttura, chiaramente quello no.
Anche una review, vabbè, autore, comunque anche gli altri hanno
Ora,
sì.
la tua qual è curriculum partiti?
Allora,
Va bene, allora se potete gentilmente mettervi qui davanti, sono 2 sedie di troppo così, ma non è un problema. Va bene, oggi tocca a me fare il discorso del Presidente, sarò breve. Per noi avere degli studenti di dottorato è davvero una grande opportunità, è un grande piacere, anche a volte un onere, è una cosa difficile, è una cosa, voi vi sarete trovati in vicinanza della fine del progetto a dormire poco di notte pensando a cosa posso riuscire a combinare da qui alla fine del triennio, ma questo vi assicuro succede anche a noi. Infatti stavamo parlando della necessità che ciascun PI abbia tempo sufficiente per seguire adeguatamente i propri studenti, perché è una cosa importante, è una cosa che io reputo sacrale, avere un dottorando di ricerca e partecipare, oltre che imparare tantissimo dal proprio dottorando, partecipare alla sua formazione. Vi ringraziamo tantissimo per questa mattinata, vi ringraziamo per averci adesso preparati come se questo fosse il vero esame. In realtà il vero esame è quello che avete sostenuto con la commissione d'esame internazionale, eccetera, ma ho visto che vi siete preparati religiosamente e questo lo apprezzo davvero molto. È un ennesimo gesto di serietà professionale da parte vostra. Abbiamo cercato di fare tutto in un paese del tutto disfunzionale, questa è la mia personale opinione, perché questa rimanesse una bella esperienza, un'esperienza del poter volentieri, anche se faticosa e anche se impegnativa. Speriamo di esserci riusciti. E quindi passo alla proclamazione. Nel nome della legge e per l'autorità conferitami dal magnifico Rettore, proclamo dottori in medicina molecolare Gabriele Antonarelli, Filippo Birocchi, Martino Alfredo Cappelluti, Valentina Pieri e Riccardo Sirtori e infine Tiziana Tempo. Le mie più sentite congratulazioni a tutti voi e grazie ancora. Ci rivedremo probabilmente da queste parti.
Va bene, ringraziamo il Presidente.
Grazie Presidente.
Bisogna mettere la crocetta per non passare.